Okay, so today I'm going to talk about mast cell activation syndrome, and this is known as MCAS. So mast cells, they're named allergy cells because they're responsible for immediate allergic reactions, causing the allergic symptoms by releasing mediators. And mediators are a protein made in response to a specific stimulus. So mast cells work with both innate and adaptive immunity, which means they're both a general response and a response for specific antigens. And they act to degranulate the mast cell, which is the process of releasing molecules from the secretory vesicles here, which results in inflammation. So mast cells are from myeloid lineage, so they had originated from bone marrow. And in this picture, we can see that the mast cells are activated upon the immunoglobulin E receptors, or the IgE mediated antigen stimulation, and releases the mediators, such as histamine, as a mediator to trigger the inflammation response. These receptors are high affinity and they're positively regulated by immunoglobulin E, and we can see the histamine degranulation coming from these histamine granules. So this picture shows the degranulation of a mediator, which is probably histamine, due to the antigen binding on the antibody on the receptor. So the exact cause is unknown, so it's idiopathic, but we can still study the physiology of the messengers, like histamine. So the mast cell likely becomes activation sensitive, overreacting to what the cell sees as an antigen. And this is likely resulting in mastocytosis, which is an abnormal accumulation or abnormal activation of the mast cells. So this can be where mast cells build up under the skin, in the bones, and in the intestines. Stressors such as the sympathetic nervous system, our fight or flight system, encourages neuropeptide release, which is small chains of amino acids that bind to the mast cell receptor that makes this degranulation. So here we can see two main receptor types. On the left, there's these, and this is toll-like receptors. And then on the right, we have immunoglobulin E receptors, where the antigen binds to the molecule, making a cross-lining. And a cross-lining is just chemically joining the molecules by covalent bonds. So there's two disorder types. There's clonal and non-clonal. In clonal, we know the evidence of the mutation and we know why. And non-clonal is when no specific abnormality has been found yet. Exogenous and endogenous histamine both play roles in the dietary physiology of MCAS. So exogenous histamine is impacted by DAO reduction, and DAO is an enzyme that the body makes to break down histamine. So we don't know the effects of humans in this very well, but we use copper in lab rats to serve as a way to degranulate the histamine, which is shown to likely degrade activation of mast cells. Again, that's in rats, that's not in humans right now. So mast cells are typically activated in the GI tract through FcRI receptors. And these receptors are high affinity immunoglobulin receptors. And we talked about DAO earlier. It breaks down histamine and results in a histamine sensitivity. So, let's see. Oops. Yeah, so eating histamine releasing foods can exacerbate symptoms due to the unhydrolyzed foods that increase MCAS. So instead, to help this, a diet of hydrolyzed foods can reduce allergen load, and FODMAP carbs can reduce carbohydrates. And FODMAP carbs are a group of foods that are good for MCAS and other things. So these carbs can reduce the carbohydrates and help the GI microbiome. That is MCAS. Thank you so much. I'll see you later.